We also talked last class about trying to determine the genotype of an egg, an oocyte, by determining the genotypes or haplotypes of all of the other cells, the polar bodies that are generated when the oocyte is produced. Does anybody have any lingering doubts, questions, concerns about this topic? So before we get into Mendelian genetics, which is the topic for today, remember I told you we're going to go into a little bit more depth and maybe a little bit more theory behind this than you might have experienced in other biology classes. So I hope you don't tune out. But before we get there, I want to talk briefly about the quiz you just took. Some of you are just finishing up. So these are the concepts we're going to try to tackle by today. The blue is all vocabulary that I hope everybody will be familiar with. Genotypes, phenotypes. What's Mendel's first law? Segregation of blank. Mendel's first law. Segregation of something. Normally it's referred to as Mendel's first law, the law of segregation. Segregation of what? You can't just have segregation. Divide something. So is it traits? We'll see. And we're going to talk about all of these vocabulary terms as well. So it looks like we've got a lot of divisiveness among the class in terms of the answers of the quiz questions, which is not surprising. So thank you for your feedback. I appreciate it. Okay. This is a really important insight into how biology works. Which direction does the arrow go? I'm going to draw an arrow. I swear I'm going to do it. Which direction do I draw the arrow? Does genotype produce our phenotype, or does our phenotype produce our genotype? The central dogma says DNA makes RNA makes protein. So DNA encodes molecules that are RNA. That's the process of transcription that we'll talk about later in the term. Then RNAs encode the amino acid language. So RNAs encode sequences that produce proteins. That's translation. And it's often sort of generally suggested that it's our proteins that give us our phenotypes. Proteins are often enzymes. They're things that do biochemistry in our cells. Proteins are responsible for skin pigmentation, hair texture, a lot of visible phenotypes, a lot of our traits. Something that's really cool about genetics, though, is that the information doesn't all have to flow to the right. Can anybody think of a case where we use phenotype to figure something out about a genotype? Can it work both ways, in other words? Can we take phenotypic information and determine something genetic? Or is it all just one-way traffic? DNA makes RNA makes protein. Our genotype infers our phenotype. Any thoughts? Sir?
Wouldn't like the Mendelian experiment kind of be like that? How we did the flower or the seed?
So Mendel's peas. You get a yellow plant, yellow peas. What does that tell you about the genotype of the pea plant? We'll wait and see. So in Mendel's case, you use phenotype term and genotype. How about something like this? So it's kind of hard to see in this picture. By the way, this is a lucky coincidence that I happen to use a human phenotype up here that we've already talked about today. I didn't mean to do this. So individuals that look female, that have unusually broad necks. Anybody know what syndrome this is? We already talked about it once today. This is Turner syndrome. So physicians regularly, I mean, this is what doctors do. You look at a patient, for those of you that are interested in doctoring, you don't have to be. Physicians look at patients, they collect information about phenotypes, and then they try to deduce, okay, well, you have all of the symptoms or characteristics of an individual that has this genetic trait. Absence of an X chromosome. So in this case, in this type of an example, a physician can actually practically diagnose an individual genotype, lack of an X chromosome, based on what they look like. So that's taking phenotype and guessing or inferring something about genotype. So the information can go both ways. Bless everybody who's sneezing. Maybe we should shut the door. I know I'm going to be a teary, runny-nosed mess in a couple of weeks after these plants continue blooming. Stupid plants and their sexual reproduction. So this is one example where you can go from oh, and then there's this. Okay. What do you see in the picture on the left? Do you see it? Are you colorblind? Yeah, me too. So those of us that are colorblind don't see anything drawn in the picture on the left. Okay, but the rest of you see what? A three? A three? A three. Okay. So presumably in there are a series of colored dots that connected together make something that look like a three. To the vast majority of you, what do you see on the right side? I'm hearing 5, 3, not sure. So on the right, is a simulated version of how colorblind people see what's on the left. So to me, and presumably to other individuals who might be colorblind in here, these two pictures look identical. So for most of you, what you see on the right is how we see what's on the left. And with this phenotypic information, this is a phenotype, the ability to see a particular color. A lot of you, some of you, didn't say that the inability to taste capsaicin was a phenotype. It is a phenotype. The inability to do something can also be, it's a trait. It's something that can be measured. It's a phenotype. What that means is if you can't taste the spicy flavors, it means that your tongue cells are not producing a protein that binds to and detects that chemical, capsaicin. It's a phenotype. Colorblindness is a phenotype. And from this information, I can tell for myself, I can diagnose my genetic condition. I'm a mutant. I have a mutation on a gene that's on my X chromosome that lets me see and distinguish red and green colors. So just from looking at that picture, I can diagnose my genotype. Such is the power of genetics. So in Mendelian genetics, we study a couple of different things. I'm not going to go through the P's, the traits, all of that. You've had that before, presumably. If not, you've read the chapter. These are the key points. If you're not familiar with these yet, I suggest coming to talk to me in office hours, reading the textbook more heavily to specifically read to understand these points, and maybe then come talk to me in office hours or separately. Okay. So these were things that Mendel, before anybody knew anything about DNA, Mendel was pre-double helix. He was pre-microscopy. No one had ever actually seen DNA or chromosomes before. Just by crossing plants and looking at the offspring of the progeny of the plants, pea flower position, the flower color, the seed color, whether or not the pods were wrinkly or round. I know I promised I wouldn't do this, but I'm going to anyway, etc. He guessed these things that are all true. So he inferred the fact that there are genes. He didn't call them genes, but he inferred that there were these units, this information that flowed from individual to individual that was necessary for making peas one color or another. to pick on the one trait of whether or not you make yellow peas or green peas. He also inferred that every plant had 2 copies of that information. Could be 2 versions. So you could either have the big Y that's involved in making yellow peas and a little y in this case that could be used for making green peas. If you combine together points 2 and 3, what do you get? What's the conclusion? What are the possible combinations of those 2 versions of the pea-colored gene? What combinations can you have as an individual? If you've got 2 copies and there are 2 types of information, 2 versions of that information. Let's make all the possible combinations. So you have 3 different combinations that we called, what are these called? What's the genetic terminology for what I just circled up here? These are genotypes. Just a simple word that means a text description of the information that's contained in an individual. I'm an individual. My sex chromosome genotype is XY. I have 1 X chromosome and 1 Y chromosome. A pea plant that makes green, let's see, does anybody remember which of these 3 genotypes makes green peas? I think it's the little y over little y. So now we're going from genotype to phenotype. If you know something about the genes, the versions of the genes that an individual has, you can usually predict something about what they're going to look like. That's the central dogma, DNA to RNA to protein. The reason we started last class with meiosis is the most amazing thing about Mendel is the bottom point. Without knowing anything, not only just about chromosomes or DNA or the hereditary material in general, he guessed truly how meiosis works. He didn't even know about meiosis. That if you have 2 versions of a gene, 2 alleles, the word allele being used for version of a gene, big Y, little y are different alleles, then exactly half of the gametes produced by meiosis will have 1 version of the gene, 1 allele, and the other half would have the other. That's Mendel's first law, point 5. That the 2 copies of a gene you have divide practically in half, well, exactly in half. Half of the gametes produced by that cell get 1 version of the gene, the other half get the other. Segregation of alleles. So that was what would have gone into that blank a few slides back. Mendel's first law, the segregation of alleles. It's how the different versions of chromosomes segregate into haploid gametes after meiosis. Questions or concerns, particularly regarding terminology? Then I'm going to stop yapping and we're going to do something. I've given you enough information, I think, to make use of some of this. I really, though, want to make sure before we do that everybody's clear on alleles, because this is one of the more confusing points, at least it was for me when I was in your seat. So this is not the phrase that I put into Google Images to get this result, although it might produce a similar result. What's an allele? Can somebody briefly, I've already defined it once, but in your own words, how would you describe what an allele is?
Different form of a gene.
A different form of a gene. A version of a gene, 2 distinct versions of a gene. So we're talking about 1 gene, but different individuals have different versions. Like I have a broken opsin gene that prevents me from seeing colors that you can see, most of you can see. So I have a different allele of that gene than you do. How many alleles could there be for any gene? Is there a limit? So I'm seeing a few tentative head shakes, which is good. How many alleles can there be in an individual human? of a gene?
Two.
2, because
We're diploid.
we're diploid. So every one of us can only have 2 alleles of every gene, but that doesn't mean that there aren't other alleles out there in the human population of the same gene. So there could be thousands of alleles of a gene, but only each individual human in this case, in this example, can only have 2 of those however many alleles there are that exist in the world. So I'm trying to dispel the common misconception that there are only 2 alleles of a gene. In practice, when we do problems in class, we'll usually only talk about 2 alleles. It doesn't mean that more don't exist. So here's a little bit of nomenclature. Here's one way to name alleles. So here are 2 different gene X versions. What's different about them? 2 different sequences call it gene X. They're both gene X. What's different? The top one's my favorite sequence, tag a cat. It's about the longest thing I can think to spell with 4 letters. Mental pictures of people spray painting felines. What's different between it and the gene sequence beneath it? Yeah, there's one nucleotide that's changed. And that's why, for example, gene X might be called X version C, X sub C. The C allele, the C version, the version of that gene that contains a C in it, at the point where in the other allele there's instead a T, so XT. It's one way to name different alleles. The point being, alleles are defined by, in some way or another, DNA sequence. When we talk about a different version of a gene or a different addition of a gene, we know they're different because their DNA sequences are different. That's the critical point. So anytime there's an allele, it's going to be called an allele because there's a DNA sequence difference between versions of the same gene. We can also name alleles based on the phenotypes they produce. So in Mendel's case, you could have a capital Y and a lowercase y. 2 different versions of the gene that causes P's to be different colors. Everybody remembers why capital and lowercase are used in this case? Typically, capital and lowercase are used in what case, or to denote what? Dominance. And we'll talk more about that in detail. So often, not always, but often when we use capital and lowercase, that's to indicate dominance relationships. And I sure hope this is the last example. Sometimes we use numbers to indicate different alleles. So this could just be gene X version 1, gene X version 2. Just add a serial number. Every time you find a new version, a new sequence of the same gene, you just add a number, X3, X4. How is this nomenclature on the bottom working? We'll see this nomenclature with the parentheses and the subscripts a few times at least during class this semester. So it's worth going over how these alleles were named. This has something to do with how police do DNA fingerprinting and forensic analysis. So what does GA2 and GA3 mean? Can you figure it out? Yeah, I think I screwed up though, didn't I? So there's a stretch there of 3 units of the dinucleotide GA. So that should actually be called the GA3. How many copies of GA are on the bottom version? There's 4 GAs in a row, GA, 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 GA. So that would actually be called the GA4 allele. That's another way to name genes. When they have a simple dinucleotide or 3 nucleotide repeat in the middle of them, you can distinguish 2 different versions of the same gene based on how many copies of that simple sequence are copied and pasted next to each other in the gene. So any questions about DNA nomenclature at this point? We've talked about alleles, we've talked about genotypes, how to name alleles. put this into practice in just a second. One other important point. Let's see. Let me add something in here from your submissions before class. Human mutants. Oh, there's a good one. That works for me. Is it real or is it Photoshop? I don't know.
I'm pretty sure it's real. My book has 7 toes, so.
Okay. So, quick question. Is this mutant or wild type? Do we call having 7 toes mutant or wild type? It's okay, we're friends here. You can say it if you want to. No one's judging anybody. We consider this mutant. What's wild type for this trait? What's normal? What's common? 5 toes. So when we talk about a mutant, when we talk about wild type, that's usually a dichotomy we use in genetics. You're either mutant or you're wild type. How do you distinguish between the two? What's the definition of mutant, or what's the definition of wild type? Mutant or wild type? If I could tell you guys now. This is mutant, right? How do you define what wild type is?
Is it based on frequency, so it's the most frequent naturally occurring trait?
Sure, it's the most frequent version of a phenotype. So, is my hair color wild type or mutant? Okay, so this illustrates an important point. That oftentimes when we talk about phenotypes, we're talking about something that's binary. We talk about Mendel's peas, they're yellow or green. We talk about whether or not you grow hair all over your body or not. Well, how much hair you grow all over your body or not. Let's talk about the number of digits that you have on your fingers and your toes. These are sort of binary traits. You've got 5 or you've got something different. You either grow hair all over your body or you don't. So there it's pretty easy to tell who's wild type and who's mutant. The most common trait is the one we refer to as wild type. If you have a rarer trait, then that's the mutant trait. Regardless of whether or not you like the term mutant and like to use it on people. For most of the class, we'll use it against animals and plants and other invertebrates, and maybe you won't feel so badly about calling things mutant. But when there are multiple versions of a trait, like hair color, it's harder to say. So often those terms aren't used for continuously varying traits, like hair color. You're really blonde, you're kind of blonde, you're dishwater blonde, you're strawberry blonde, you're brown, you're brunette, you're auburn, you're, I don't know, whatever. I'm colorblind, so I don't know all the hair color names. So to this point, hopefully this is where we are in class. We've got mutations, they change a DNA sequence, and alleles are the names we call the different DNA sequences of genes that are found in different individuals or within the same individual. And when the phenotypes are binary, mutants are the individuals that have the less common or less frequent phenotype, the one that's rarer. Everybody's okay with this so far? So we've defined genotypes, alleles, wild type versus mutant. Now we get into the definition of what we're going to learn about in Mendelian genetics. There are 2 different types of Mendelian genetics. This illustrates one of them. This is my pedigree. So it's looking at transmission. This is transmission genetics, Mendelian genetics. It's looking at the inheritance of something over generations. What do we look for in pedigrees? What do the dark symbols, the filled-in symbols indicate? We haven't done this in the chapter yet. I'm just wondering if anybody remembers.
Individuals that display a certain trait.
Right, individuals that have a certain trait. In this case, which trait did I pick in my pedigree? Color blindness. So my mom's father, my maternal grandfather is colorblind, so am I. So a transmission geneticist, a Mendelian geneticist, this is one of the things that we'll be doing in class. This is something you would do. You build a pedigree and you look at how a trait, the phenotype, passes from generation to generation. The other thing we're interested in is looking at how DNA passes from generation to generation. So we're looking at inheritance of DNA over time, and we're looking at the inheritance of traits, phenotypes, over time. So genealogy represents phenotypes and how they pass from generation to generation. Your favorite genetic tool of all time, the Punnett square, represents looking at how alleles segregate and distribute themselves from generation to generation. So in essence, the Punnett square and the pedigree are two different sides of the same coin. One looks at the inheritance of traits over time, one looks at the inheritance of genes over time, from generation to generation. So for example, for my pedigree, we saw that that trait skipped a generation. My grandfather had it, nobody in the next generation had it, and then I have it. Here, another version of Mendelian genetics lets me guess, infer, that my mom is the one that gave me my mutation. Thanks, Mom. We know that because it's on her X chromosome, and she got that from her father. So we'll be doing a lot of Punnett squares in the immediate future, and after that we'll be talking about pedigree analysis. Right. Now you all should be able to do this based on what we covered last class. So here's a diploid cell. Draw what it looks like as it goes through meiosis. So what's going to be the first step? I'll walk you through the first couple of steps, then I'll leave you to your own devices. So here's a diploid cell. 2N. It's got two copies of the same chromosome. What are big Y and little y? We'd call those different alleles. So I've color-coded, in this case, you don't have to. I've color-coded the two versions of the same chromosome, blue and red. If that cell is going to undergo meiosis, what's the first thing it's going to do? It's going to replicate. So we have to do S phase, synthesis. Okay. So what letters, let's forget drawing sticks to represent chromosomes. Let's just use the alleles. What's present in that one cell after synthesis? How many letters and which flavors, which varieties? So each of the initial letters duplicates.
2 big Y's and 2 little Y's.
So we've got 2 big Y's, the original one in the copy. We've got 2 little Y's, the original one and the newly made copy, essentially. Okay, now it's your turn. So draw what that temporarily tetraploid, right, 4 copies of one chromosome, draw what happens after the first meiotic division and the second meiotic division. Anybody remember how the first division works? What do we put in the top cell up there? Okay, so it's the 2 versions, the 2 alleles that came from the same parent, the original and its copy, go together in the first division. That makes things somewhat easy, right? What happens now? Second division, what goes in every gamete? One letter. So we've gone from a 2N cell that's diploid to 4 times N, or haploid. So each of those cells has N, 1 copy of each chromosome. They're haploid. What's the genotype of the cell we started with? How would you write the genotype of the cell that we started with, the diploid cell, the blue and the red one? If you're diploid, how many letters are there in your genotype? 2. Right, it's the genotype, the written representation of the alleles found at a given locus. So the slash is critical. The slash represents the separation between 2 different molecules of DNA. So on one side of the slash, we have one allele. On the other side of the slash, we represent whatever the allele nomenclature is for the other version of that gene. That's a genotype. Where on the slide are haplotypes? It's another really critical piece of nomenclature. If there are 2 things you remember that's vocabulary, I hope it's genotype and haplotype. They're very closely related, they're just different. What does the word haplotype kind of sound like? That's already up there on the screen. Haploid. So a haplotype is the genotype of a haploid cell. Okay, so what's the haplotype of this cell? It's big Y. No slash needed because there's only one piece of DNA that contains that gene. There's one chromosome that has the Y on it. So the haplotype of that cell, you would just write as big Y. Right. Genotype has the slash because it indicates the separation between 2 chromosomes, 2 pieces of DNA. So that's the difference between genotypes and haplotypes. Genotypes, if you're talking about a diploid, you'll have 2 letters. So let's see, which of the following, this is from the quiz, which of the following represent genotypes of a diploid organism? About 20, maybe 10% of you, 15% got that one right. So is any nomenclature, the choices were big A over little a, big B over big B, plus over minus. This is a different question. I'm just going to run you through both at the same time. Which of those represent genotypes? This is a new question. This is not one of the questions I asked on the quiz. All of them. Okay, so they're all genotypes. Options that were not genotypes on the quiz were single A or a single B, for example, not genotypes. Which of these genotypes are heterozygous? Here's terminology we haven't used before. Heterozygous. The A's and the H's, and the plus and the minus. Though a heterozygote being any individual that has 2 different alleles in one individual. Hetero, different. So if you have 2 different versions of a gene, you're a heterozygote. If you have 2 same versions, you're homozygote. You make the same zygotes, same gametes. The gametes all have the same haplotypes. Yeah, Google Images is wonderful. He's a purebred dog. Come on. Next time I'll show you a picture of an inbred cat. It's true, I will, but it's funny. You can imagine what it looks like. So we'll talk about, again, pure breeding individuals and those that are not. So pure breeding individuals, the definition is you mate two individuals together and they have, all of their offspring have the same trait. So we focus on any one trait at a time. Say you start with two yellow pea plants, you cross them together. If all of their offspring only make yellow peas, then we know that the original parents were pure breeding. They produce a pure next generation in that genotype, or in that phenotype rather, pure. So what sort of a genotype, now we're talking DNA, what sort of genotypes will produce pure breeding individuals? Would you be heterozygous or homozygous? Right. So for example, if you have an individual that's homozygous for the big H, and you mate that individual with somebody that's got the same genotype, this is the test for pure breeding. Mate them with an individual that has the same genotype, also big H over big H. Which generation? Now we're setting up crosses for the first time, so I have to be careful. What do we name that first generation in a cross? It's not just because of Mendel. P stands for something else. Is the P0 generation? Stands for parental generation. So anytime we set up a cross, the first generation of the cross we'll call the P0 parental generation cross. And I know that most of you know this because most of the class answered this correctly on the quiz at the start. When one of these diploid cells undergoes meiosis, what are the gametes going to contain? That individual has only big H's. So what is the version of the chromosome that goes into its gametes? Has to be big H. There's no other choice, right? All of its gametes have to be, so what do we call that? The genotype of a gamete? Haplotype is big H. And the same is true for the other parent. So the P0 gametes can only have big H's because that's the genotypes. There is no heterozygosity. So what happens when we combine this sperm with that egg? We create an F1 individual that has to be the big H over big H genotype. That looks pure breeding. We start with two parents that have the same genotype. We wind up with 100%, that's the critical part, 100% of all of their offspring, not just this one. Every time those two parents mate, I guarantee you they make HH genotype offspring. So 100% of any possible offspring those two parents would have are going to have that genotype. Therefore, this is pure breeding. What's the difference between what we see here and what happens when you're not pure breeding? What changes? What genetically makes individuals be not pure breeding? Right, they're not homozygous. They must be heterozygous. So we could do the same thing, but now what's the heterozygote genotype? Let's say small h. So again, if we're trying to find out if this individual with this genotype, big H over small h, is pure breeding, what are we going to cross this individual to? Who? What genotype? We're going to use the same genotype. That was the P0 generation. So parental generation, what do their F1s look like? Maybe we should back up a step. What's in the gametes of the P0 individuals? If you draw out meiosis for each one of those individuals. This is what we did at the start of class. Take a diploid cell, make it go through meiosis, draw me the gametes. What do these look like? Right. Mendel's first law, segregation of alleles. You start out with a heterozygote. An individual can only have 2 copies, a diploid individual can only have 2 versions of the chromosome. In this case, they're heterozygous, big H, little h. That means after meiosis has to be the case that exactly half of the gametes have big H's and exactly half have little h's. That's the only way the math works out. Start with 4 chromosomes, divide them into 4 separate cells, 2 big H's, 2 little h's. And the same over here. So that's through the process of meiosis. Now what do we have to do to predict, because this is Mendelian genetics, it's all about predicting, watching chromosomes pass from generation to generation, predicting what's going to happen when you set up a cross. What do you predict the F1 are going to look like? Here we have the gametes of 2 individuals. How do they combine together to produce the next generation? What sort of genotypes can we get in the F1 generation? I'll start off for you. I'll pick a big H from the parent on the left. Now what? What gamete gets used from the parent on the right? Could be another big H. Okay, what else could happen?
Homozygous recessive, so 2 small h's.
So we could get 2 small h's pairing together. That would give us a little h over little h. We've got a big H over big H combination there. And then in red, if we happen to have a little h from one parent and a big H from the other, you'd get the heterozygote combination. It should all be mostly review, or at least starting to sound familiar again. What just happened? This is the last thing that's important for appreciating Mendel's first law, and then next class his second law. What did we just do up here with these arrows? What did I do? What did you describe to me? Is there any combination we didn't use of gametes on the left and gametes on the right? It's like we took the gonad of parent on the left and emptied out all of its gametes. We took the gonad from the parent on the right and emptied all out its gametes and just smushed them together, and they fertilized. Right? All the sperm, all the eggs, all together, made all the possible combinations, right? What do you describe that process as? What one word? Fertilization, in this case, is blank with respect to which haplotypes? It's random. Right? It doesn't matter whether or not the parent on the left contributed a big H or a little h to the offspring. That choice is totally independent of what happens on the right side. So big H's and big H's can fertilize, big H's and little H's and big H's, little H's and little H's. It's the last important concept for Mendelian genetics. Law 1. Fertilization is random. So we're going to skip this horrible looking big table. You can do it if you'd like to. I think it would make an important point. That's why I put it in here, but we don't have time for it right now. So I suggest, we've already done the top. We've already filled in this part. At the start of class, I asked you to take a cell, draw what it looks like after meiosis, then look at what happens if you do 2 cells, what happens in meiosis after you look at 4 cells, and then if you really got time, try doing this 1,000 more times. I kid, of course. Do the same thing for heterozygotes. What does meiosis look like after you do meiosis from just a single heterozygous diploid cell? Then what does it look like when you do 2 separately, 4 separately? So this is something I would like you to do that makes exactly the same point. So don't write anything. Maybe letters, just to indicate haplotypes. draw a picture or diagram, make a diagram that accurately represents the number of different genotypes, that is haplotypes, produced in all of the gametes produced by one heterozygous individual over its entire lifetime. Draw me that in the blank space here, post a screenshot of that to this Google Classroom site. So you follow one individual, doesn't matter, mouse, rat, cat, plant, over its whole lifetime. It's a diploid individual, it's heterozygous, big A over little a. Draw me some sort of diagram, don't write, draw a description of how many big A gametes and how many little a gametes it produces over its entire life. And then, so do that by next class. This is my other request. We haven't talked about setting up Punnett squares yet, but hopefully you brought some of this information with you or you read about it in the book. Here's our first Punnett square to set up. So draw a diagram of Punnett square that represents what would happen in the next generation if you cross those individuals together. And then we'll have all sorts of Punnett square fun, and I promise you will learn something about Punnett squares that you never knew before. It is not historical either.